So, guys, gals, let's talk about the skeletal system. When we look at our skeletal system, this first presentation is going to be about 2 class periods. We're going to focus on a lot of the physiology of the skeletal system today and on Wednesday. Friday of this week, and Monday, Wednesday of next week, we're going to focus on the axial skeleton, the appendicular skeleton, and then joints and classifying movements. So there'll be a lot of overlap between lecture and lab starting Friday through Wednesday of next week. When we look at bones, guys, gals, these are often thought of as very static tissues and that they don't change that much. But that's not necessarily the most accurate thing. They are constantly changing. They just don't change as fast as some of the other tissues of our body. For instance, when we think of our epithelial tissues, our skin, the lining of our stomach, those tissues are rapidly changing. Every 30 days, we replace our skin on our epidermis. Every 3 days, we replace the lining of our stomach. Those tissues have rapid turnovers. When we think of our blood has a turnover of about 4 months. Every 4 months, you replace all the red blood cells of your body. Our bones aren't replaced that quickly. It takes about 10-ish years to replace all the tissue of your skeleton. You're constantly refreshing your skeleton, just not at the same rate that you refresh the other tissues of your body. When we look at bones, there's a root word, osteo. Osteo is a root word for bone. So when we think of osteology, that's going to be classified as the study of bones. Now, as we look at our bones, we need to make sure we're very clear about something. All of our bones, most of the bones of our bodies, in particular the long bones of our body, started out as cartilage. As a rule of thumb in our bodies, cartilage is the precursor before bone. I know we have a couple wrestlers in here, and a common condition that happens for wrestlers is cauliflower ear, where they tear the elastic cartilage in their ears, they get a blood clot, the blood clot turns into cartilage, and then the cartilage can solidify and occasionally turn into bone. And individuals that have very bad cauliflower ear will have extra bony tissue forming in their ears. It's really kind of a weird looking phenomenon. I remember when I started to develop cauliflower ear, I had to have the blood clots drained every morning and every night with a big syringe in my dad's kitchen. When we think of ligaments and tendons, they are dense, regular connective tissues. And on a microscope, they look very similar to each other. The distinguishing characteristic of a ligament and tendon is what they're attached to. Tendons are going to have a skeletal muscle on one side and a bone on the other side. So they attach muscles to bone. Ligaments, on the other hand, are going to have bone-to-bone connections. Now, as we look at our skeletons, they do a lot of things for us. Some of them are really intuitive. You know, they give us support. They give us protection. I mean, those most people can come off with tacitly just thinking about it. Some of the other functions of our skeleton are a little bit less intuitive. For instance, we use our skeletal system to maintain the correct pH in our body, our acid-base balance in our bodies. We have 2 chemicals in our bones that we use to regulate pH. We have carbonates, and we also have phosphates. And these carbonates and phosphates that are present in our skeletal system are dumped into the bloodstream, and they act as a buffering system. They can absorb hydrogen ions, or they can release hydrogen ions. They act as an acid and a base and resist changes in the pH of our blood. We also use our bones to make blood. In our bones, there's a special kind of bone tissue called spongy bone tissue, or cancellous bone tissue for the old school name. And in that spongy bone tissue, we have red bone marrow. And this red bone marrow is where we make the blood in our bodies. So we use our bones also to make blood. Now, as we look at the tissue that is present in our bones, we have lots of different kinds of tissues. We have connective tissues, we have epithelial tissues, and we also have nervous tissues present. Our connective tissues in our bone are kind of unique connective tissues in that they are the only tissue in our body that is a solid at room temperature. That makes it unique in that it's the only tissue in our body that is a solid at room temperature. We'll have osteoblasts, bone-building cells that secrete collagen, and then those collagen fibers start to have crystals form around them. And those crystals are going to be the hard component of our bone. And they aren't like big and sparkly and shiny crystals. They're itty-bitty microscopic crystals. The process of adding crystals to the collagen in the extracellular matrix, or the process of making the bones hard, is called mineralization or ossification. Those are going to be synonyms with each other. To mineralize or to ossify is to add the inorganic, the mineral component to the collagen and make the extracellular matrix rigid. When we look at a bone, we can consider a bone both a tissue and an organ. It all depends on the frame of reference. If we look at compact bone, that's a tissue. Spongy bone is a tissue. The femur is an organ. So think of the femur, the longest bone in the body, the thigh bone. That femur is going to have dense regular connective tissues, compact bone tissues, spongy bone tissues. It's going to have adipose, loose connective tissues, and it will have red bone marrow. There are 5 different tissue types in our femur. Those multiple tissue types are working together for a common purpose, and that's the definition of an organ. So a bone can be an organ, or we could have bone tissue. It all depends on your frame of reference and how the question is asked. As we look at the bones in our bodies, we can categorize them based on their shapes. The most easily recognizable bone in our body is going to be the long bone. And when we think of the long bone, those long bones are, longer than they are wide. Long bones are, generally speaking, going to be in the arms, legs, or your ribs for the thoracic cage. As we look at long bones, because they're longer than they are wide, they serve as excellent leverage points in our body. And we'll have type 1, type 2, and type 3 levers present with our long bones. This is not a class on biokinetics, so we're not going to talk about torque calculations or how to define the fulcrum versus the applied or resistant forces. But when you think of a long bone, I just want you to think of them as a lever that can maximize either your torque or the force being generated, or can maximize the movement that's being produced. When we think of a short bone, it has approximately the same length and width. So in other words, they're very small, roundish type bones. Our short bones are going to be prominently located in the hands, the carpals of the hands, or the tarsals of the feet. And these short bones aren't necessarily going to be for leverage or levering action, but instead are going to be present where we want just a little tiny bit of movement. Think about how your foot, the bones of your feet, can spread out just a little bit when you take a step versus when you stand up or remove the force. We have, question, comment. There are multiple classifications. So we have a lot of short bones, we have a lot of long bones. Let's go to the next slide. Irregular bone is the miscellaneous category for our bones. We have flat bones as well. And when we think of flat bones, those are bones that are thin, they form plates. Classic examples of flat bones include the bones of the cranium, so the frontal, the parietal, the temporal, the occipital bones. The sternum is another classic flat bone, or you could have the ilium and ischium of the pelvic girdle. Those are flat bones as well. So when we think of a flat bone, I want you to think of something that protects underlying soft tissues or underlying organs. So we'll have long bones, we'll have short bones, we'll have flat bones, and then there's all the bones that are really hard to categorize. That miscellaneous category is called irregular bones. Classic examples of irregular bones include the bones of the face. I mean, there's not a consistent shape to the bones of our face, or the vertebrae as well, being classic examples of irregular bones. So let's have a concept check, guys, gals. The blank is an example of bone as an organ, and blank bone is an example of bone as a tissue. So we have about 30 seconds left. Remember, an organ is multiple tissue types working together for a common purpose. And think of those tissues that I mentioned verbally at the beginning of this presentation. We're down to about 2 seconds. Let's go ahead and check our results. The correct answer is femur is the example of bone as an organ, and spongy bone tissue is bone as a tissue. So it looks like we have the right idea. A couple of us had some minor dyslexia and flip-flopped femur and spongy bone tissue. Let's talk about some of those tissues in more detail. We have compact bone tissue. We've already covered that in lab 2 weeks ago. Compact bone tissue is also known as dense bone tissue. Sometimes it's also referred to as ground bone tissue. Ground bone tissue is a really old school term. Pretty much the only time you ever see that is when you're looking at really old microscope slides in lab. They'll say ground bone cross-section on them. And that refers to dense bone or compact bone. Another kind of bone tissue that we see present is spongy bone, also known as cancellarius. And there's a really old school term for this called trabecular bone. And it's named after the individual little slivers or the individual trabeculae that make up the spongy bone or trabecular bone tissue. That term has kind of fallen out of favor. Spongy bone is my favorite name for this, and it's the most common name. It's being used in all the new textbooks because it's a very descriptive name. It looks like a sponge. Now, when we look at compact bone tissue, every bone in our body is going to have compact bone tissue around the outer perimeter. And then in the inside, we're either going to have a hollow space or we'll have some spongy bone tissue. In terms of mass, most of the mass of our skeleton is going to be that dense or compact bone tissue. As its name implies, it has a lot of matter or material put into it. The spongy bone tissue isn't necessarily for structural support, but primarily focuses on as a framework to hold red bone marrow. Although, yes, there is some structural support from the spongy bone tissue. Now, as we look at a generic long bone, we have the middle portion and the end portions. The middle portion of a generic long bone is known as the diaphysis, and the end portions, the proximal and distal portion of a long bone, is known as the epiphyses. When we think of the epiphyses, there are going to be lots of anchoring points on the epiphyses. Lots of tendons and ligaments will, generally speaking, be attached to the epiphyses of the bone. Yes, there are exceptions, but as a rule of thumb, tendons and ligaments will attach to the epiphyses. When we look at the diaphysis, it serves two primary purposes. Number one, it increases the length of the lever of the long bone. And then number two, it's the hollow part of the bone that serves to store additional bone marrow. That hollow part of the diaphysis is known as the medullary cavity. And I'll admit, I'm a little crusty now. I'm getting kind of old. So in my mind, it's just been beat into me as the medullary cavity. Although your textbook, which is being revised with more current terms, refers to it as the marrow cavity within the diaphysis. And the marrow cavity is a better name. It's more descriptive. It describes what that hollow space does for us. It stores marrow. Now, to separate the diaphysis from the epiphyses, there is literally a line that we can use. This line is called the epiphyseal plate. Most of you probably know of this as the growth plate. And if you are still growing taller, your growth plate is going to be made of hyaline cartilage. When you stop growing taller, the growth plate loses all of its hyaline cartilage. And instead of being categorized as an epiphyseal plate, it'll be recategorized as an epiphyseal line. So let's talk application here. One of the primary reasons why breaking a bone at the growth plate or the epiphyseal plate is so detrimental to an individual is because during the healing process, that hyaline cartilage that made up the epiphyseal plate will be replaced with compact bone tissue. In other words, the individual will stop lengthening the bone at the site of the breakage if they break their bone at their growth plate. When we stop growing taller, if we look at where the growth plate was, the epiphyseal line is going to be a line of compact bone tissue. And I'll highlight that for us right now. So there'll be a line of compact bone tissue that separates the diaphysis from the epiphyses. So in this room, we have a bunch of people, young, healthy people in this room. So you will have a big, bold, beautiful epiphyseal line if there's an x-ray taken of your long bones. For geriatric patients, as we age and we go through that constant bone remodeling, the epiphyseal line starts to blur, and the compact bone tissue that made up the epiphyseal line starts to be replaced with spongy bone tissue. And the loss of that compact bone tissue at the epiphyse is one of the reasons why geriatric patients have so many bone breakages. Think of how common it is for someone to fall and break their femur if they're aged, but if they're young, generally speaking, their femur is going to be okay. They might get a bruise, but they'll be all right. At the proximal and distal end of every long bone in the body, and present on a lot of the short bones of the body, we have articular cartilage. Now, if you look at the histology of articular cartilage, it's just hyaline cartilage. It's hyaline cartilage at a joint or articulation point. In lab this week, we have articulated skeletons. We have articulated plastic skeletons. We also have 4 articulated real human skeletons for you to work with. They're super cool. And we also have disarticulated skeletons, so skeletons in a box with all the bones disconnected from each other. As we're looking at this articular cartilage, it serves a very important purpose. It reduces friction. The articular cartilage, or hyaline cartilage present in the joints, reduces friction. It makes it easier to move our joints. Individuals that develop arthritis usually start to lose their articular cartilage. They lose the lubrication in their joints, and they have a lot of extra friction and inflammation of their joints. In order to supply nutrients to keep our bones alive, we need a point for the blood vessels to go in and out of our bones. And the hole the blood vessels go in and out of are known as nutrient foramina. I struggle with that word. And this nutrient foramina, or foramen, as I was taught, is generally speaking just a little hole in the bone. There are some foramen that are very large that you're going to have to memorize in lab, such as the mental foramen that are present on the chin. Other foramen are so small and vary from person to person to person that they're not worth committing to memory. So from person to person, there are large common foramen that are present on everyone, and then each of us have a smattering of itty-bitty tiny foramen that vary from individual to individual. We also are going to have a membrane that lines the medullary or marrow cavity, and that membrane is known as the endosteum. Endo meaning in, oste or osto for a bone. It's the inside membrane of a bone. And the endosteum serves a very important purpose. Think of a small child's femur versus an adult's femur. The small child's femur has a much smaller medullary cavity than the adult's femur will. And to enlarge the medullary cavity, the endosteum has a lot of cells present that dissolve bone tissue. And as the endosteum dissolves compact bone tissue, it will enlarge the medullary cavity to allow for more marrow to be stored. We also have a membrane around the outer perimeter of the bone, known as the periosteum. The endosteum dissolves compact bone tissue. The periosteum adds compact bone tissue. So as our bones are growing wider, we'll have the periosteum add more compact bone tissue to the outside of the bone. This periosteum can add compact bone tissue throughout our entire lifetimes. Sometimes we'll have a hormonal cause. There'll be individuals that produce too much human growth hormone. That could result in gigantism or agronomalise. Gigantism is what I call it. It's easier to say. Other times, you can change your physical routine to stimulate more compact bone deposition. If you go from being a couch potato to having a lot of resistance training, exposing your bones to lots of torsion and forces and resistance will stimulate the periosteum to lay down more compact bone tissue. In our periosteum, to lay down this extra compact bone tissue, we have a lining of cells that specializes in laying down the compact bone tissue. And the periosteum serves two purposes. It makes our bones thicker. It also allows for tendons and ligaments to be attached to our bones. Let's talk about a generic flat bone. So the last several slides have focused on a generic long bone. When we look at a generic flat bone, we're going to have the dense bone tissue on the inner surface and on the outer surface. And then in between the two layers of the compact bone tissue, we'll have spongy bone tissue. So you can think of a flat bone as a spongy bone sandwich. The spongy bone is sandwiched in between two layers of compact bone tissue. Because our flat bone has lots of spongy bone tissue, our flat bone will also have lots of red bone marrow stored in its spongy bone tissue. I'm going to say that again because that's a key component of maintaining homeostasis. We as adults will have red bone marrow present inside of our flat bones, and we need that red bone marrow to make new blood cells. This is the site of blood cell generation for us as adults. With children and infants, red bone marrow is much more common because they need to generate a lot more blood. But as adults, we aren't increasing in volume rapidly, so we don't need as much red bone marrow. So let's have a concept check. A long bone is covered externally with a sheath called the blink, whereas the marrow cavity is lined with another sheath known as blink. So what are the two membranes associated with long bones? We have one around the outer perimeter and one on the inner lining that lines the medullary cavity, or marrow cavity, to use the new term. We have about 15 seconds left. So, guys, gals, that membrane on the outside of a long bone is the periosteum. The membrane on the inside, around the marrow cavity, is the endosteum. And it looks like we have a pretty good concept of that, or grasp of that concept. Let's move on and talk about the cells of bone tissue. We're going to have adult stem cells, or multipotent stem cells, that make our bone tissues. The specific kind of multipotent stem cell that makes bone cells is called an osteogenic cell. Osteo meaning bone, and if we look at genic, the root word there is for genesis or creation. And I always think of the first book of the Old Testament that has the story of creation. Genesis in Latin means to create. So an osteogenic cell is a cell that creates other bone cells. And these osteogenic cells are going to be found in the endosteum and the periosteum, those two membranes that wrap around long bones. Depending on the forces the bone is exposed to, your dietary situation, or the age of life, where you are in your life cycle, that'll influence whether you produce different kinds of bone cells. So let's talk about one of the most popular ones, an osteoblast. Think of a couple weeks ago when we talked about fibroblasts. A fibroblast is a fiber building cell. We also talked about chondroblast, a cartilage building cell. When we think of an osteoblast, I want you to think of a bone builder. The blast for to build, osteo for bone. We have osteoblasts present in both the periosteum and the endosteum. We can make our bones thicker from the inside or from the outside. And these osteoblasts make new dense bone tissue a very specific way. First, they secrete collagen fibers. After those collagen fibers are secreted, they will serve as the nucleation site for crystal formation, as the mineral that makes up the extracellular matrix will precipitate out of solution and form that rigid component of our bones. Now, if we expose our bones to lots of stress, we're going to stimulate osteoblast activity. The example that always comes to my mind is of a professional baseball pitcher where they have their dominant arm they throw with. They're constantly stressing out the bones of that arm, and they'll have dramatically increased bone density in the dominant arm versus their non-dominant arm. Another example that kind of strikes close to home for me is with my younger brother. He studied martial arts for a while, and one of the things his sensei taught him to do was to punch a tree. And I remember I made a lot of fun of him for going outside and punching a tree repeatedly. But after a while, it paid off. His bone density of his carpals and his metacarpals was dramatically increased. And after punching a tree for a couple of years, he could punch me really hard. Those osteoblasts, as they secrete extracellular matrix, will bury themselves in extracellular matrix. And once they're completely encapsulated in the extracellular matrix, they shift gears and they morph from being an osteoblast to an osteocyte. An osteocyte maintains our bone tissue. And as we look at a traditional or typical osteocyte, it'll have lots of tiny little branches coming off of it. Those tiny little branches are called canaliculi. And they will serve as the stress indicators. As we stress or flex our bones, the caniculi of the osteocyte are going to sense that distortion, that stressing of our bones, and will cause the cytoplasm to flow in and out of the caniculi as they're squeezed by the compact bone tissue. And as we have the caniculi squeezing and relaxing and having cytoplasm flow in and out of the main body or cell body of the osteocyte, that will stimulate the osteocyte to secrete a little bit of extra matrix to make the bone just a little bit stiffer. Now, this osteocyte needs some room to sit in. So we call that space the osteocyte sits in the lacuna. It's a hollow cavity that's present in the compact bone's extracellular matrix. We also have osteoclasts. An osteoclast serves to destroy compact bone tissue. And yes, it's very important that we have cells that destroy our bones in our bodies. This is all part of maintaining homeostasis. We need to have the correct concentration of calcium ions and phosphate ions in our blood. If we have too many, our osteoblasts will make our bones thicker. If we don't have enough calcium or phosphate in our blood, the osteoclasts dissolve our bones a little bit and dump calcium and dump phosphate into our bloodstream. Or let's think of as we're growing up or growing larger. As we go from infancy to childhood to adolescence to adulthood, and the medullary cavities increase in volume, we need osteoclasts to dissolve that compact bone tissue so that the medullary cavity or marrow cavity can become larger. So there's some give and take. Something that's notable about osteoclasts is that they are one of the very few cells of our body that are multinucleate. Up until this point, every single cell that we've talked about in this class has been mononucleate, with the exception of skeletal muscle tissue and some cardiac muscle cells that are binucleate. Now, as we look at these osteoclasts, they dissolve our bone tissue by secreting a very specific chemical. They secrete hydrochloric acid underneath the periosteum or underneath the endosteum, and that hydrochloric acid will dissolve the calcium and phosphates that make up the extracellular matrix so that they can dump the calcium and phosphate ions into our bloodstream. Let's talk about the extracellular matrices of our bone. So we've been talking about how we need to maintain calcium and phosphate ion concentrations in our body, and we use the osteoblasts and osteoclasts to add or subtract to the extracellular matrix. Let's talk about that extracellular matrix in more detail. There's a very specific name for the mineral that makes up most of our extracellular matrix. It's calcium hydroxyapatite. And this calcium hydroxyapatite makes up 85%, or the overwhelming majority of the volume of our compact bone tissue. We're also going to have some calcium carbonate and then some miscellaneous chemicals as well. Because our compact bone tissue is relatively inert, one of the advantages of compact bone tissue is we can use it to sequester chemicals in our bodies that we want to get rid of. So individuals that have suffered from lead poisoning, mercury poisoning, cadmium poisoning, individuals that have had heavy metal poisoning will store those extra heavy metal ions in the matrix of their compact bone tissue to remove those ions from the circulation so that they don't affect the proteins or carbohydrates of their bodies. But primarily, our extracellular matrix is going to be calcium ions and phosphate ions. And there's two parts to our extracellular matrix. We have the collagen fibers, the organic component, and we have the calcium hydroxyapatite, the inorganic component. You could think of the collagen and the mineral as rebar and concrete. There's been a lot of bridge construction in this region recently, and you may have noticed that when there's a lot of construction projects, first the iron workers will put down rebar, and rebar is flexible and can bend, and then around that flexible rebar they pour stiff and rigid concrete, and that makes the buildings, generally speaking, pretty strong. Our bones have this same property. This rebar concrete construction method mimics our bones. In our bones, the collagen, or the organic component, that collagen protein fiber, gives us flexibility to our bones. And then that calcium hydroxyapatite and the calcium carbonate, those minerals, make our bones stiff. Individuals that don't have enough mineral in their bone are going to have flexible, bendy bones. Individuals that don't have enough protein or collagen in their bones are going to have very brittle bones. And you can do this at home. This is a fun experiment to do. When I was a high school teacher, I'd have my students do this in a lab setting. They would take one bone and put it in a beaker full of vinegar and let it sit there overnight. And then he'd come back the next day, and that bone was super bendable. They could wrap it around their fingers after soaking in vinegar overnight. And then they'd also take another bone, and they'd put it in, we'd have a drying oven in my classroom, and we'd bake the bone at 400 degrees Fahrenheit for about a school day, about 8 hours. And after 8 hours at 400 Fahrenheit, the bone would be super brittle, and the students could squeeze the bone in their hand and have it turn into fragments. So let's have a concept check. The hardness of bone comes from blank, where blank provides some degree of flexibility. So we'll stop at the 30-second mark. So we'll have about 20 seconds left. So ask yourself, what is that most common mineral present in the bone? And then what is that what is the organic component of our bone? So as we look at this, hydroxyapatite and the calcium carbonate and those other miscellaneous minerals make our bones stiff. And then the protein, specifically the collagen, is going to make our bones flexible. But there is more than just collagen present in our bones. Let's talk about compact bone tissue. As we look at compact bone tissue, you're going to find a lot of overlap between what you covered in lab 2 weeks ago and what we're covering in lecture right now. As we look at compact bone tissue, they have a lot of osteons present in them. And I need to camp on osteons for a while. These osteons, or concentric rings of our compact bone tissue, are the structural and functional unit of our compact bone. Each layer of the osteon is called a lamella. At the very center of the osteon, we have the central canal, also known as the Haversian canal. That's the old school name. And then within our osteons, we have some sideways passageways called perforating canals. We use the central canal of the osteon to have an artery, a vein, and a nerve travel through the compact bone tissue. And then those transverse canals will also have an artery, vein, and nerve. So we use these passageways through the compact bone tissue to deliver nutrients, remove waste products, and provide innervation, provide nervous system connections to our bones. We also have a lot of spongy bone tissue. As we look at spongy bone tissue, that spongy bone tissue is going to primarily be located in the epiphyses of our long bones and in our flat bones. So spongy bone tissue is in the epiphyses of long bone and in flat bones. The individual fibers within spongy bone tissue are referred to as trabeculae. And as we look at this photomicrograph, the trabeculae are the pink portions. So I'll outline that in green. The trabecula is the bright pink portion. And then there's that red spotted or red mottled material. That red mottled material is actually going to be red bone marrow. And there was a lot of red bone marrow present on our pubic symphysis slides that we were looking at 2 weeks ago during lab. On those slides, you were focusing primarily on the fibrocartilage of the pubic symphysis. But I remember as I was bouncing around and helping students, a lot of individuals were using their cell phones to take pictures of the red bone marrow present on that slide. Now, our spongy bone tissue isn't going to have a lot of osteons. Compact bone tissue has tons of osteons present in it. But because our spongy bone tissue needs room for the red bone marrow, and just to emphasize it again, red bone marrow makes our blood. That spongy bone tissue won't have the physical space for the osteons that the compact bone tissue does. We find also that our spongy bone tissue, it's a nice compromise between strength and weight. Yes, our spongy bone tissue is not as strong as compact bone tissue, but the advantage of spongy bone tissue is it's a heck of a lot lighter. If we weren't concerned about mass, we go with just compact bone tissue. But there is a certain point where it becomes so heavy it's not worth it. So we use compact bone tissue around the outer perimeter when we really need that strength. But to fill the hollow spaces where we still need some strength, but we don't want a lot of mass, we will use spongy bone tissue. And as we look at the trabeculae of our spongy bone tissue, the lines of the trabeculae mimic the forces that the bone is exposed to. And these trabeculae remodel over our lifetime. We've looked at fetal bone, infant bone, and adolescent bone. And as that child goes from kind of the larval stage, I call it the 4th trimester, to crawling, to walking, to running, they expose their bones to different forces. And the trabeculae are going to reorient based on the different forces the bone is exposed to at their different stages in the life cycle. In our skeleton, for an adult skeleton, we'll have red bone marrow present in our spongy bone tissue. Most of our spongy bone tissue is going to be located in our flat bones or the epiphyses of our long bones. So primarily in our axial skeleton is where we'll have red bone marrow as adults. All the other medullary cavities present in an adult skeleton are going to be filled with yellow bone marrow. And yellow bone marrow has a very specific name. It's adipose loose connective tissue, or fat, for lack of a better term. So we have the Easter holiday or the Seder meal coming up. And depending on your family tradition, you'll either have maybe a ham or a pot roast, and you'll have that big bone present in it. The reason we'd like to take those bones and make soup out of them is because the yellow marrow, or the fat that's present in the medullary cavity, really adds nice flavor to the soup. I personally am a big fan of split pea soup with a ham bone. Now, as we look at an infant or a child, they need to generate a lot more blood. So in children and infants, they have almost no yellow bone marrow. In children and infants, it's almost exclusively red bone marrow. And as we age, our red bone marrow is progressively replaced with yellow bone marrow. Or another way to think of it is, our ability to make new blood cells is replaced with fat that we're storing. So I think this will be a good time to call it quits for today. We'll answer this last review question, and then we'll call it quits for the lecture. What would you find in the marrow cavity of the diaphysis of an adult humerus? Adult being a key term here, not infant or child. Would you have a periosteum, hematopoietic tissues, red bone marrow, yellow bone marrow, or compact bone? Think back to the last slide we just covered. We use red bone marrow to make blood. It's a hemopoietic tissue. Yellow bone marrow is also known as fat. Let's go with 10 more seconds. The correct answer was yellow bone marrow. Well done, guys. I'll see you in lab. Have a great Monday.